you see anabolism. We're making big from small. We're making polymers from monomers. We got to put energy in. Catabolism. We're breaking down big molecules into smaller molecules. Digestion in our body. When we digest food, we take large things in, break them down into small things we can absorb. And that's all catabolism. And that releases energy. So we're taking polymers and making monomers, and we're giving off energy and we're giving off heat. And dehydration synthesis. It's how we make polysaccharides. What's the storage form of sugar in animals? Storage form of sugar. Not what we turn into when we run out of room. Because it's not lipids. Lipids are not carbohydrates.
That's what I was thinking.
Storage form of carbohydrate. Lipid is not a carb. Lipids are lipids. How about glycogen? Something we got to know. Stored in the liver and skeletal muscle. We're going to hear that a dozen times before the end of 2 semesters. In plants, what is it? You all know it. No. What kind of a vegetable is a potato? Starch. Yeah, starchy vegetable. Starch. What's cellulose? An example of fiber, undigestible carbohydrate. It is a polysaccharide, but it is not a storage form. It is fiber. There are different forms of fiber and types of fiber. Cellulose is the example we use because it's in the cell wall of plants. No, it is not part of any animal I ever knew. Because animals can't absorb fiber, and we don't make fiber. But we do use fiber in our digestive tract to help us what? Stay regular, not have our blood sugar go way up and spike, and what else?
To poop right.
Well, I said for regularity. How about cholesterol and coronary artery disease? To the Cheerios box. Those things are important to know down the road. I mean, triglycerides are glycerol and 3 fatty acids. So that's the most common form of lipid in our body, but lipids aren't the building blocks. Fatty acids are. Proteins, the building blocks are the amino acids. And we put them together and we make larger molecules. There you see the 3 fatty acids, the building blocks. A triglyceride. Here you see 2 amino acids. When we put a bunch of these guys together and we get a whole string of them, we get what? A polypeptide, and then amino acid chain in that sequence is what? The primary structure of the protein. Catabolism is breaking down. It's the opposite, hydrolysis. We put water in to break the bond. So yeah, it is the reverse. We use water to break apart those complex carbs, that glycogen and starch that we consume, to break down the lipids and as well as the proteins. We're going to learn. You get the AMP2. Remember, what is the, I told you this, but see if you remember, what is the suffix for enzymes? We'll talk about it here in a few minutes. How about ACE, ASE? OSE is sugar. ACE is enzyme. ASE. So if it ends in ASE, it's an enzyme. So if we had a protease, what do you think it breaks down? Proteins. If it were a peptidase, it would break down peptides, which are several amino acids put together. Break it down into the amino acids. We'll talk about that in AMP2. We'll talk about lipases. Break down lipids. Go ahead.
I have a weird question on that. So peptides are just amino acids put together?
Put together. Yeah, polypeptide chain is what that amino acid chain is that's the primary structure. A peptide is like 10 of them put together.
So
a dipeptide is like 2 peptides put together. A whole bunch of peptides put together is that amino acid chain that's the primary structure of the protein. Go ahead.
Oh no, I was just wondering because like in my my vitamin stuff, it says amino acid, or not my vitamins, my proteins.
Yeah.
I take it says peptides, but it also says amino acids.
Yeah, and peptides are amino acids put together, like say about 10 of them. A polypeptide is a chain of like say 100. So you're putting a bunch of peptides together. And the polypeptide chain, when we're done making it, is the primary structure of a protein. See, when we talk about translation, the actual making of the protein from the information we got from the DNA. We'll do that. We'll talk about that next week. And it's just the reverse of making that dipeptide. We're breaking it down into the amino acids. We're breaking down triglyceride into glycerol into 3 fatty acids. Now, these have to occur in sequences. Okay, so we have a bunch of these metabolic reactions. And enzymes, remember, they are catalysts. And everybody say, well, they speed up reactions. Well, that's what protein catalyst means. They're proteins and they speed up a reaction. And how do they speed up a reaction? They don't stop it from going. They make it run. But how do enzymes speed up a chemical reaction? What do they do? We talked about it. We'll talk about it again right now.
They lower the activation.
Yeah, the energy of activation. They lower the energy of activation. They make it easier for these guys to run, and they are specific. As you will see here, one reaction, one enzyme. They are a fit for each other. And that fit is known as an induced fit. Induced. What does it mean to induce something, ladies? I'm picking on the ladies for a reason now. Lisa, last time I checked, you and I never went into labor.
To hurry up, to push out, induce.
To get it going, yeah, like inducing labor. That's why I picked on the ladies, because I know I haven't, and I doubt if you've ever gone into labor.
No, not yet.
No. That'd be a news story.
Hey, that Danny DeVito movie.
That'd be a news story there. But yeah, to induce labor is to get it to go. And in this case, what induces the fit are coenzymes, vitamins. Coenzymes, which are vitamins. Cofactors, which are minerals. Coenzymes, vitamins. Cofactors, minerals. And here is the discussion on the enzymes.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex.
Okay. Now this is an example of what kind of a reaction? What would this be an example of? We got 2 reactants here. So what do you think we're doing? Putting them together. So this is going to be an example of dehydration synthesis. Enzymes can equally work in the opposite way. Enzymes can just as readily work in the opposite way. And they form an enzyme-substrate complex. And this, again, is that induced fit with the courtesy of the coenzymes, the vitamins, cofactors, the minerals, and they form a complex. And at the end, this enzyme is neither used up nor altered. It is unaltered at the end, and it's not used up. So let's take a look at the rest of this.
The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the 2 substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from 2 substrate molecules, other enzymes catalyze the formation of 2 products from a single substrate.
In other words, the reverse. Now, enzymes, they lower the energy of activation. So yeah, they control reactions. Most of them, yeah, they're globular. They're all proteins. They're mostly globular. Yes, they all have specific shapes. They are not used up, they are not altered. They are specific to each substrate. One substrate, either the single molecule or the multiple, when you're putting things together, they are specific to that substrate. So one chemical reaction, one enzyme. It is substrate specific. And the shape of the active site determines the shape. Remember, they have to fit. Try putting the square peg in the proverbial round hole. Does that work very well? Does it work very well to put a round peg or a square peg in a triangular hole? Not exactly. And that's what we've got right here. We need the round peg for the round hole, the square peg for the square hole, the pentagonal shaped peg for the pentagonal shaped hole. One substrate